Good day everyone. For today's topic, we'll be discussing on the Coccidians. These are the references for this lecture. Now, the Coccidians belong to the phylum Apicomplexa. They are characterized to be spore-forming microscopic single-cell parasites. They are considered to be an obligate intracellular protozoans, which means that they need host cell to reproduce. Primarily, they infect the gastrointestinal tract. They usually cause severe diarrhea, and the stool is considered as the diagnostic specimen of choice. The confirmatory diagnostic modality to diagnose coccidians is through stool culture. Coccidian infection results to a condition called coccidiosis. It is host specific, except for Toxoplasma gondii, which can infect all mammals. Coccidian infection is considered opportunistic in immunocompromised hosts. They are common in HIV/AIDS patients and transplant patients in malnourished children. It may be asymptomatic in immunocompetent individual, meaning it doesn't result in any symptoms if you have a healthy immune system. The members of coccidian infection includes the Isospora, Cyclospora, Cryptosporidium, Toxoplasma, and Sarcocystis. The life cycle of coccidian has this a pattern of alternation of sexual and asexual reproduction. It occurs in three sequential stages: gametogeny, which is the stage where it produces a micro and a macro gametocytes or gametes. Second is through sexual cycle or sporogony, producing oocyst containing sporozoites. Third is asexual cycle or schizogony, producing merozoites or also known as merons. So the coccidians are considered to be one of the complex protozoal infections because of their life cycle. But if you understand their life cycle in terms of the sequential stages and what products they produce, you would understand its biology. So as you can see here, the typical life cycle of coccidia, we have again the gametogony, sporogony, and schizogony. The first parasite that we will be discussing is the Cytoisospora or Isospora bellii. For this parasite, humans are the only known host. Transmission is through the ingestion of mature oocyst, also known as the sporulated oocyst, containing two sporocysts with each containing sporozoids. So as you see here in the illustration, the entire thing is the mature oocyst, or also known as the sporulated oocyst. It contains two sporocysts inside, and each of the sporocysts has a 4 sporozoid. Sporulation of oocyst occurs in food or water. We can find the Isospora bellii in the distal duodenum and proximal ileum. This is the life cycle of Isospora bellii or Cystoisospora bellii. So first, if humans usually get infected with ingestion of mature oocyst with sporozoites, and then it infects man to the gastrointestinal tract, wherein it is converted into wherein the mature oocyst releases sporozoites and undergoes an asexual phase, or also known as the schizogony. Now it produces merozoite, and these merozoite undergoes differentiation to microgametes and macrogametes and undergoes sexual reproduction phase. And this is also known, the sexual reproduction phase again is also known as the sporogony. Now, sporogony results into the oocyst containing two sporoblasts. Now, asexual development occurs in one host. The asexual or schizogony life cycle, the process is where you have a mature oocyst containing two sporocysts, each with 4 sporozoids. When ingested, release sporozoid invading intestinal cells of the intestine, forming trophozoites and eventually merozoites. Then the sexual reproduction or sporogamy. Some merozoites develop into a macro or microgametocyte, resulting to zygote, then forming an oocyst, which are then passed out with feces and undergo maturation in the environment. Generally, if a healthy person is infected with isospora belli, it would manifest as an asymptomatic infection. The disease is more common in children and in patients with HIV/AIDS, especially those with immunocompromised, those with cancers, and organ transplant patients. Cystoisosporiasis is a prolonged mild diarrhea, sometimes lasting for 6 months, and associated with abdominal pain, fever, malaise, flatulence, malabsorption syndrome with weight loss, and dissemination of parasites to other organs in AIDS patients. In terms of the morphology of the oocyst of Isospora belli, we have an immature oocyst, which is elongated and ovoidal. See here in the first illustration in the left side. Second is we have immature oocyst with 2 sporoblasts. And third one is the mature oocyst with a sporocyst, and each sporocyst contains 4 merozoites. The diagnosis of Isospora belli is the visualization of oocyst in fecal specimen through DFS or concentration technique. You can also perform acid-fast staining, and acid-fast staining is commonly used in tuberculosis, but it can also be used in diagnosing Coccidian infections. Enterotest and duodenal aspirate can be done, and usually we can suggest the diagnosis if we can find eosinophilia and Charcot-Leyden crystals in stools. Now, Charcot-Leyden crystals, these are the byproduct of the metabolism that occurs inside eosinophils. Okay, so in the enterotest, here in the enterotest, the patient will swallow a capsule, and the goal of this test is to get samples from the jejunum or duodenum. And this is eosinophilia, the presence of eosinophils in blood, and Charcot-Leyden crystals. This is a typical illustration of the sporocyst, and it is the mature oocyst that contains 2 sporocysts, and each of it contains 4 sporozoites. That is the infective stage. So any of these can be found in stool specimen. Okay, so you can see here, you can see the in a biopsy specimen of Isospora belli. Treatment is trimethoprim sulfamethoxazole, pyrimethamine, and sulfadiazine. We prevent this infection through food and water precaution, proper human waste disposal, and proper hygiene. Next is Cyclospora cayetanensis. Originally, it is a cyanobacterium-like body. They have like photosynthesizing organelles and autofluorescing particles characteristic of blue-green algae. Transmission is the ingestion of mature oocyst, and the mature oocyst contains 2 sporocysts with 2 sporozoites each. This is in contrast to that of Isospora belli, in which it contains 2 sporocysts with 4 sporozoites, but for Cyclospora cayetanensis, it contains 2 sporocysts with 2 sporozoites. Please remember that. The habitat of Cyclospora cayetanensis is in the small intestine, with jejunum as its predilection. So the mechanism of the disease lies in the life cycle of the parasite. We have the ingestion of sporulated oocyst. Again, each oocyst contains 2 sporocysts with 2 sporozoites each. The oocyst release sporozoites in the intestine, and then the sporozoite invade the epithelium of the small intestine, preferably jejunum. Then sporozoites undergo merogony. Again, when you say merogony, it is the asexual reproduction. It is divided into 2 generations. The first generation, the merons, contains 8 to 12 merozoites, and the second generation, the merons, contains 4 merozoites. Now, some merozoites undergo gametogony to form micro and microgametes. The micro and microgametes then fertilize and initiates the sexual reproduction or the sporogony, and now it forms an oocyst. So the microgametes again fertilizes macrogametes, forming an unsporulated oocyst passing out with feces. The oocysts undergo a complete sporulation in warm environment for 7 to 12 days. Okay, so I think I explained this one in the illustration. So the pathogenesis of this parasite is generally self-limiting. When we say self-limiting, our immune system takes care of it to just go away. The initial symptoms of this infection of Cyclospora cayetanensis infection is patient usually presents with malaise or body weakness, low-grade fever, and it can potentially result into chronic intermittent watery diarrhea alternating with constipation. And the diarrhea would last for 6 to 7 weeks with having more than 6 stools per day. So if a patient presents with chronic diarrhea, one of your differential diagnoses is Cyclospora cayetanensis infection. Other symptoms would manifest as fatigue, anorexia. Anorexia means the patient has low appetite, low weight loss, nausea, vomiting, abdominal pain, flatulence, bloating, and dyspnea. Now, in Cyclospora cayetanensis, it's very unique for them that we have a D-xylose malabsorption. Now, D-xylose is a type of carbohydrate. It is quite an uncommon carbohydrate. It is typically involved in pentose metabolism, but in Cyclospora cayetanensis, this is a unique infection in which it results into D-xylose malabsorption because it affects the proximal small intestine malabsorption. There are no deaths reported for this infection. Diagnosis is through direct microscopic examination. Concentration techniques through acid-fast staining. The oocyst, very typical of this one, is the autofluorescence, and the fluorescent microscopy would result into blue or green circles. We can also perform polymerase chain reaction to verify this infection. So as you can see here, the illustration showing you the blue-green fluorescence. Again, Cyclospora cayetanensis is originally a cyanobacterium-like body. The treatment is not necessary if symptoms are mild because they are self-limiting. But if they result into moderate to severe symptoms, then we can treat with trimethoprim, sulfamethoxazole, and it is the only proven drug if treatment is warranted. Prevention and control is through good and sanitary practices, adequate water treatment such as boiling water. Chlorination is not effective. Proper washing of fruits and vegetables should be observed. Next is Cryptosporidium. It is an opportunistic infection of man. When we say opportunistic, the parasite would result into manifestation of the disease if the immune system of the host is low. It is an intracellular extracytoplasmic parasite. They have a predilection to the brush border of our enterocytes, and transmission is through the ingestion of thick-walled oocysts from the environment, pets, and farm animals. Habitat is in the gastrointestinal tract. We have two important species. We have Cryptosporidium hominis and Cryptosporidium parvum. All stages of development are completed in the gastrointestinal tract of the host. So here you see these parasites here. These are the Cryptosporidium. This is the enterocytes. And you see here the Cryptosporidium, they have a predilection in the brush borders or the apical surface of the enterocytes. Now for their life cycle, humans pass out thick-walled oocysts. The oocyst is already mature upon passing out, and when ingested, the oocyst releases 4 sporozoites, which infects intestinal cells. Now, the sporozoites undergo schizogony to produce merozoite type 1, which results into 8 daughter cells. Okay, so again, if an infected human passes out thick-walled oocyst, it now results into contamination to the environment, especially in the drinking water, and humans infect is infected by this parasite by ingestion of thick-walled oocyst, and this thick-walled oocyst contains 4 sporozoites. Now, it is different from all the other parasite because it doesn't have a sporocysts in each. Now, it has only the sporozoites. Now, that sporozoite, when it enters the gastrointestinal tract, it undergoes schizogony, or again the asexual cycle, and it now produces meronts, type 1 meronts, and that type 1 meronts forms merozoite, and that merozoite undergoes type 2 meront, which will result into sporogony. The type 2 meront undergoes sporogony. Okay? So this is what I've discussed. Some merozoite will infect other intestinal cells to produce merozoites type 2, and some merozoites become microgametes and macrogametes through gametogony to form oocyst. The thin-walled oocyst immediately releases sporozoites, which results into autoinfection. So here we have autoinfection, meaning it will propagate more of their parasite and infecting other neighboring enterocytes. The thick-walled oocyst is then excreted through the feces. Okay, so this is the life cycle, and this condition that results from this parasite is what we call cryptosporidiosis. For immunocompetent individual, it would manifest as asymptomatic or just a self-limited diarrhea, which is the most common symptoms, and some symptoms that may present are pain, anorexia, fever, nausea, and weight loss. For immunocompromised individuals, patients may have severe chronic diarrhea, such as an AIDS patient. They are also at risk children in daycare centers, animal handlers, healthcare workers. And for immunocompromised patients, they have diarrhea of more than 2 weeks, dehydration, abdominal pain, anorexia, fever, nausea, vomiting, weight loss, and malabsorption. And why do we have malabsorption? It's because these parasites infects the brush border. And what is the usual, or what is the function of our brush border? Our brush border contains a lot of enzymes, a lot of proteins, a lot of transporters that carry out absorption of nutrients such as carbohydrates and proteins. So what happens if we are infected with this infection? We have blunted intestinal villi, resulting to malabsorption and fluid loss. For extraintestinal, we have heavy infection of bile duct and gallbladder, and it may result into acute gangrenous cholecystitis or the inflammation of our gallbladder. For pulmonary cryptosporidiosis, we can have dyspnea, chronic cough, bronchiolitis, and pneumonia, and possibly conjunctiva. The oocyst of Cryptosporidium contains 4 crescent-shaped sporozoites, and it's the infective stage. Now, this is unlike to other Coccidians, in which they have a sporocytes or sporocysts. Here, they don't have a sporocyst, but it's directly a sporozoite. Diagnostic methods: you can perform a Schietter's sugar flotation technique, the formalin ether ethyl acetate concentration technique, or FECT, the modified Kinnean's acid fast staining, and very typical of this staining is the appearance of red-pink donut-shaped circular organism in a blue background. And lastly is intestinal biopsy, and you can find here evidence of Cryptosporidium and the brush borders. So this is the brush border. Again, it is intracellular but extracytoplasmic. It's outside the cytoplasm, but it's still intracellularly. So there's a border of plasma membrane around it. So we treat it with hydration and symptomatic treatment. Nitazoxanide effective among immunocompetent patients, but unclear efficacy for immunocompromised patients. There are promising treatments such as bovine colostrum, paromomycin, and clarithromycin. One important thing about Cryptosporidium is that chlorination does not affect oocyst, and we can prevent it through waterborne transmission control. We have to have proper hygiene and boiling or filtering of water. Next parasite is the Sarcocystis. It infects humans and animals worldwide. The definitive hosts are humans and carnivores. The intermediate hosts are herbivores and omnivores. And the humans serve as definitive host for two species. We have Sarcocystis hominis and Sarcocystis sui hominis. This is the life cycle of Sarcocystis. The sporulated oocysts and sporocysts are discharged in the feces. And the intermediate host, usually it's an herbivore. The oocysts or sporocysts are ingested by herbivore, then sporozoites penetrate into the intestinal mucosa, then into the blood vessels. The sporozoites undergo schizogony to form merozoites, and some merozoites penetrate the muscles and form the sarcocysts. Now, when I say sarco, it is related to muscle. Sarcocyst. That's why it's called sarcocystis, because it has something to do with the deposition of the oocysts to the muscles in herbivores. The sarcocyst in muscle of herbivores contains bradyzoites, and the meat is eaten by human, usually through raw or undercooked. And the sarcocyst present in that muscle releases bradyzoid in the intestines, and the bradyzoids penetrate the intestinal mucosa and becomes a microgamete and macrogamete. Now, the bradyzoid is somewhat similar to the merozoites. It is just a unique name. They're called bradyzoids because they have a slow reproduction rate. Now, the microgametes and macrogametes form two oocysts with two sporocysts, and each of the sporocysts has 4 sporozoites. Now, this is typical or has a similar morphology as that of the first one, Isospora belli. This is the life cycle of the parasite. Basically, they have the same. But what is unique about Sarcocystis is we have an ingestion of meat containing containing oocyst, and that oocyst contains a lot of bradyzoids. Now, that bradyzoids is eaten by man, and the bradyzoids infect the intestinal mucosa, and then in the intestines there's a formation of sporocyst containing sporozoites. Okay, this is the life cycle of Sarcocystis for the asexual phase or the schizogony. It occurs in the intermediate hosts such as herbivores or such as pigs or cows. The sporulated oocyst containing 2 sporocysts and sporocysts containing 4 sporozoites is ingested by the intermediate host such as cows or pigs. The sporozoid in gut epithelium infects the blood vessels through the endothelium, and then it undergoes schizogony. The second and third generation merozoites it occurs in the mononucleate cells, and the third generation merozoites, or also known as the multinucleated schizont, results into a single non-infection metrocytes and insist in the muscle. And it now results into the asexual reproduction of metrocytes and now forms mature infectious bradyzoids, or also known as the sarcocyst. Sexual reproduction or sporogony occurs in the definitive host such as man, and sarcocyst contains bradyzoid that is ingested by man, and the wall is digested and the active bradyzoid enter the intestinal cells, and the microgamete and microgametes then form and eventually fertilize, and it now forms a zygote, and this zygote will result into oocyst containing 2 sporocysts containing 4 sporozoites each, and it's passed through the feces. Okay, this is the typical appearance of the mature oocyst of Sarcocystis. You see here that it is an oval transparent consisting of 2 mature sporocysts with 10 to 18 microns. Each sporocyst contains 4 teardrop-shaped sporozoites. Here, teardrop-shaped sporozoites. Now, this illustration shows you this first one. You have the first generation schizont inside the endothelium, and this occurs in the intermediate host. This is the second generation schizont found inside the monocyte. This also occurs in our intermediate host, such as the herbivores. The third generation schizont is inside the glomerulus, and the third generation schizont is found usually in the muscles. Diagnosis of sarcocystis is fecal flotation wet mount using bright field microscopy. The definitive diagnosis is the biopsy of the infected muscle using hematoxylin and eosin stain. For Sarcocystis hominis, it is the microscopic muscle of the cattle. For Sarcocystis sui hominis, it is the uh, microscopic, um, uh, macroscopic, macroscopic examination in the muscle of the pig. For periodic acid shift, it will result into a positive wall stain, and we can also perform PCR. Okay. So, what's the manifestation or the clinical manifestation of this disease? It is usually asymptomatic and clear spontaneously. The disease that is caused by this infection is the sarcosporidiosis or sarcocystosis. We have the intestinal form resulting from the ingestion of sarcocyst containing bradyzoid. We have the muscular form, which is the ingestion of oocyst or sporocyst. Now, this table shows you the parasite development and disease manifestation in humans. And you can see here the characteristic and in the result in the intestinal infection. The source of infection of sarcocystis is usually from raw or undercooked meat. The infective stage is the sarcocyst containing bradyzoids, the developmental stages. Here we have the sexual stages in the lamina propria of the intestines, and the oocyst is excreted in feces. The time to ingestion of infective stage to symptoms is around 3 to 6 hours, lasting 36 hours. Symptoms include nausea, loss of appetite, vomiting, stomach ache, bloating, diarrhea, dyspnea, and tachycardia. Diagnosis is through demonstration of oocyst or sporocyst in feces beginning 5 to 12 days after ingestion. We have no approved therapy for this one. Okay. For intestinal type, we have no treatment, but for muscular, we can treat through albendazole, metronidazole, trimethoprim, sulfamethoxazole, or a steroid. Prevention is thorough cooking or freezing of meat to kill bradyzoid, boiling water, and unfortunately, we don't have any vaccines for this one. Next is Toxoplasma gondii. Toxoplasma occurs worldwide. It infects humans and many species of animals. The disease is manifested in immunocompromised individual. The definitive host is the cat family. The intermediate host is other animals like mouse, bird, pigs, cows, and sheep. Humans act as accidental host. The definitive host of this infection is the cat family. The unsporulated oocyst is shed. It undergoes sporulation in the environment. The sporulated oocyst is ingested by the intermediate host such as the mouse or bird. So this you see here, the definitive, when you say definitive host again, it is where the parasite undergoes sexual reproduction or attains sexual maturity. So therefore, it is also the stage where oocysts are formed, because again, oocysts are the result, or oocysts are a result of sexual reproduction. So as you can see here, this illustration shows you the cat passes out fecal oocyst, and the typical life cycle of the toxoplasma is that oocyst is passed through the intermediate host such as mouse and bird, and here uh, they undergo um, formation of tissue cysts such as tachyzoids, and then that tachyzoid is now eaten by cat, and the cat again is being infected by the same parasite over and over again. But accidental host accidental host such as humans can be infected through first and be through contamination of food, litter box, or water containing fecal oocyst, or The fecal oocyst infects other animals such as pigs or sheep, wherein it forms tissue cyst, and that tissue cyst is ingested by human. Another is through blood transfusion containing the parasite. Okay. So again, going back to the typical intermediate host of Toxoplasma, the tachyzoids localize in neural and muscle tissue, and it now insists to form bradyzoids. The insisted bradyzoids is consumed by cat, and the cat directly ingests sporulated oocysts. For human infection, we can get them through eating undercooked meat, consuming contaminated food or water with cat feces, and what's present in the cat feces is the fecal oocyst. Through blood transfusion or organ transplantation or transplacentally from mother to fetus. The tissue cyst or oocyst releases tachyzoids in the intestines of human, and the tachyzoids enter the blood circulation, including the fetal circulation, and it insists in neural and muscle tissues. It can insist in brain tissue and in muscle tissue. And what is found, or what form of parasite is present in these tissues? We have cyst containing bradyzoids. So the life cycle of Toxoplasma gondii is quite complicated because it entails a lot of hosts and a lot of forms of the parasite. But it is important for you to know that the cats are the definitive host. The intermediate hosts are mammals and birds, such as mouse and birds. But humans are only the accidental host, but they follow the same pattern, the same infection as that with the intermediate host. They are not the normal host for the life cycle of Toxoplasma. It just happens that we are also mammals, and they behave the same way as that to its normal intermediate host. So again, how do we become infected with parasite? It can be through ingestion of the spores. Now the spores infects the intestinal cells, releasing sporozoite. The sporozoite infects the intestinal cells. It now releases tachyzoids, and then it invades the monocyte in the blood, and then it now disseminates to other parts of the body, such as brain, heart, lung, eye, muscles, and placenta, and it forms now cyst-containing bradyzoids. And that cyst-containing bradyzoids are the form that is present in the infected tissues. We have three forms of Toxoplasma gondii. We have the oocyst, tachyzoids, and bradyzoids. The oocyst is the infective form. It has a similar appearance to those of Isospora, though smaller. It contains two spore cysts each with 4 sporozoites. The tachyzoid is a crescent shape. Has this dimension. It has a single central nucleus, and it is actively dividing form of the parasite. The bradyzoid, these are slow growing forms. When you say brady, it means slow. Tachy meaning fast. Okay, it is similar to tachyzoids, but they are smaller. We have hundreds or thousands of bradyzoids enclosed in a cyst measuring 12 to 100 microns in diameter. And again, as what I've said, the bradyzoids, these are the forms of the parasites that are found in our muscle, brain, heart, especially in humans. In a healthy individual, it results into asymptomatic infection. We have a condition called toxoplasmosis in which tissue cysts are present in human. Again, what form of parasite is found in these tissues? We have the bradyzoids. It can be deposited in the skeletal muscle, myocardium, brain, and eyes. For acute toxoplasmosis, it manifests as fatigue, lymphadenitis, or the inflammation of our lymph nodes. And why do we have inflammation of the lymph nodes? Again, there is a certain point of their life cycle wherein it infects monocytes. and monocytes are highly present in our lymph nodes. Chills, fever, headache, and myalgia or muscle pain. Chronic toxoplasmosis result into encephalomyelitis or the inflammation of the brain substance, myocarditis or the inflammation of our heart muscle, hepatitis or inflammation of our liver, retinochoroiditis or the inflammation of the blood vessels of the retina with subsequent blindness. Congenital toxoplasmosis occur if there's mother-to-child transmission. It is severe and fatal and result into hydrocephalus, microcephaly, intracellular calcification, chorioretinitis, convulsion, and psychomotor abnormalities. They may also have mental retardation, severe visual impairment, or blindness. So the classic triad of congenital toxoplasmosis is hydrocephalus, intracranial calcification, or chorioretinitis. So if you see this, it is highly suggestive of toxoplasmosis. Cerebral toxoplasmosis commonly occurs in AIDS patients. I have a condition called toxoplasmic encephalitis manifesting as headache, fever, altered mental status, lethargy, focal neurologic deficits, brain lesions, and convulsion. This is extremely common in HIV/AIDS patients here in the Philippines. If we see any neurologic symptoms in patients with HIV/AIDS, then we highly suspect toxoplasmosis. Diagnosis is through the demonstration of tachyzoids and cysts containing bradyzoids in tissues. We can also perform serologic testing using Sabin-Feldman test and IgM antibodies in congenital infection. Methylene blue can stain the cytoplasm of toxoplasma, and toxoplasma is incubated with patient's serum. If the patient has antibodies, and the complement will destroy toxoplasma membrane, thus it will not be stained with methylene blue. If the patient has no antibodies, toxoplasma remains intact and is stained by methylene blue. And this is a principle of the Sabin-Feldman test. Management is through pyrimethamine plus sulfadiazine with folinic acid. Now, again, review the mechanism of action of this one. And I want you to figure out, why do we include folinic acid? What is the folinic acid for? For pregnant women, the first 4 months, we can give spiramycin. The succeeding months, we can give the pyrimethamine plus sulfadiazine plus folinic acid. We don't want to give pyrimethamine plus sulfadiazine during the first 4 months because if we give these drugs during the first 4 months, it can result to the formation of neural tube defects in the fetus. So we don't want to result to that one. Prevention is the avoidance of contact with cat feces. Meat and eggs should be cooked well. Unpasteurized milk should be avoided. Pregnant women should avoid contact with cats, and laboratory workers should be very careful in handling parasites, especially if it contains toxoplasma. And that ends my lecture. Thank you.